Hi again, this is Mohamed. The last video we talked about, an overview steps of meiosis. So we're gonna talk about each step in this video. So where we look at closer, where we take, a closer look at each phase of the meiosis. So the first step is the prophase, in meiosis. So the first step is a prophase in meiosis, prophase 1 in meiosis 1. In this process, the chromosome begins to condense, where this, process is called synapsis. So it's a homologous synapsis. The synapsis is a homologous chromosome where each are made up of two sister chromatids. Now each chromosome is in tetra, a complex of 4 chromatids. So chromatids of homologous chromosomes are now, crisscrossed, which helps the holding of chromosomes together. This process is known as the chiasmata. So the centrosomes move away from each other, and spindle microtubules form between them. As we talked about, in mitosis, where we say centrosome moves, away from each other, and spindle microtubules form between them. It's just the same as here in meiosis. So finally, the spindle microtubules captures the kinetochore, so a protein structure on chromosome that forms the chromosomes, where the chromosomes begins to move to the metaphase plate. Important note that prophase 1 occurs more than 90% of the time required for meiosis. That's how it takes, how much space it takes. The second step is the metaphase 1 in meiosis 1. So the chromosomes are now arranged on the metaphase plate, still in homologous pair. Then the kinetochore microtubules from one pole of the cell are attached to one chromosomes of each pair, while microtubules from opposite poles are attached to the homologous pair. The third step is anaphase 1 in meiosis 1. Now the chromosomes now appears individually like different chromosomes that are attached. That's how it appears in a metaphase plate. Different chromosomes appears, or pair of different chromosomes appear. Then the spindles separate the sister chromatids of each chromosomes. The first step is the telophase and cytokinesis. 1. The spindle apparatus now continues to separate the homologous chromosomes until the chromosomes reaches the pole of the cell. Now each pole has a haploid chromosome set, but each chromosome has 2 sister chromatids still. The cytokinesis, division of cytoplasm, as we talked about in meiosis phases, occurs simultaneously in this phase, forming 2 daughter cells. Important note that in some species, chromosomes decondense nuclear membrane and nuclearly reform, forming interphase 2 before mitosis 2. So there will be no further replication of genetic materials prior to the second division of mitosis 2, which is mitosis 2. Mitosis 2, the separation of sister chromatids. So in meiosis 2, the separation of sister chromatids. This is when the separation of sister chromatids happens. In meiosis 2 of the preface 2, the spindle apparatus forms and the chromosome progresses towards the metaphase plate. So in meiosis 2, metaphase 2, the chromosomes are positioned on the metaphase plate in the mitosis within the kinetochore of sister chromatids, each chromosome pointing towards the opposite poles. In anaphase of meiosis 2, the chromosomes of sister chromatids finally separate. Then the sister chromatids of each pair now, which is individual chromosomes move towards opposite pole of the cell. We have in telophase 2 in meiosis 2. The nuclei form at the opposite pole of the cell, and cytokinesis occurs. At the completion of the cytokinesis, there are 4 daughter cells, each with a haploid number of unreplicated chromosomes. Here's again, the phases of meiosis, as we talked about everything about meiosis. So here's the picture of everything that we talked so far. Each step, what I did is I talked about each step, then put in the picture of each step that I talked about it. Origin of generation variation. How do we account genetic variation? Sexual life cycle to produce genetic variation among offspring. One, the behavior of meiosis and mitosis is mostly responsible for the variation of for the most of the variation that arises. I want to say that, not that. I'm sorry, that arises from each generation. So the explanation of these different types in the details of meiosis 1. So the two mechanisms that contributes to the genetic variation arises from sexual reproduction, and they are independent assortment and crossing over. The crossing over occurs during prophase 1 and meiosis 1 when the tetrads are formed. Chromosomes are going to line up in the homologous chromosome. Chromosomes are very close to each other in the fact that they overlap and exchange pieces in a process called crossing over. This is a process. Okay. So we have a genetics. Genetics recombination occurs in the offspring might have a combination of genetic traits that neither parents had. Independent assortment. This is when the chromosomes will line up the middle of meiosis 1 plate as a matter of chance, where each chromosome will orient itself independently of the other pair. So if you want to talk about the let's compare comparisons of meiosis and mitosis. We have mitosis occurs during the interphase before the meiosis. So for meiosis occurs during interphase before the meiosis. So in terms of number of division, one in prophase, in doubling prophase, metaphase and anaphase, and which is mitosis and telophase. So two each, including prophase, metaphase, and anaphase, and telophase. That's number of division. Synthesis of homologous does not occurs during prophase 1 in meiosis, with crossing over between non-sister chromatids, resulting chiasmata holding pair together due to the sister chromatids, cohesions. Number of in mitosis, the number of daughter cells and genetic composition, two each diploid and genetically identical to the parents. For each haploid containing half many chromosomes as a parent cell, genetically different from the parent cell and from each of other cells of each other. Role in the animal body, enables multicellular adults to arise from the zygote, produce cells for growth, repair, and some species, asexual reproduction. So produces gametes, reduces number of chromosomes by half, and introduces genetic, variability among the gametes. That's the difference between, two. That's the end of the slides. That's the end of the meiosis. It's kind of easy and interesting. How cell divides. Yeah. Thank you guys. Yeah. Bye, guys. And I hope you like this video. Subscribe and like it. Thanks.